In this video, we will discuss about mechanism of enhanced cytotoxicity of CD19-CAR-NK92. So, in the previous video, we have seen the results of this CAR and CD19-CAR-NK92 with CD19 positive target cells, and we are able to see that compared to wild type NK92, CD19 CAR-NK92 has enhanced cytotoxicities. So from the literature, they are able to find out that there are two mechanisms by which this NK92 works. First, by releasing lytic granules such as Granzyme and Perforin, and second, through this triggering through the use of this Fasl ligand that can interact with the dead cell receptors in the target cells and trigger apoptosis. So they wanted to find out which of those mechanisms plays a role towards their enhanced cytotoxicity. So they make an individual knockout of both of them and dual knockout also. And the methods they used is shown in the panel A, where first they took the CAR-NK92 cells and then transduced them with the Cas9 plasmid. And after 3 days they started the selection of this plasticity, and after 14 days they use the sgRNA, the guide RNA plasmid, and thereafter day 70s they use the neomycin selections in order to select these guide RNAs. And after week 7 they create a single cell clones and then scale them up till this week 11, where thereafter they sort the cells or check the cells for this sgRNA. At the same time they also confirm that the cells, the knockout of the respective proteins using Western blot, and those Western blot results are shown in this panel B, where we can see that in the case of this Perforin knockdown, Perforin is not shown, and in the case of Fasl knockdown, Fasl not shown, and when both of them are knocked down, both of them are not shown. They did check in the different clones that are shown here. It is 4, 5, 6 and 7, 8 and 8, 9, 10. In both the cases, the respective expressions of the other proteins are almost same. For example, in the case of Perforin knockout, the Fasl expressions are not changed significantly, and similarly for the case of Fasl knockout, the Perforin expressions are not changed significantly, but the trail expressions change, or we can say that its expression is inconsistent in the knockout compared to the wild type for both the clones, or for the different clones that they have observed. So once confirming the knockdown of this proteins, what I did is they then check the CD19 expressions to confirm that knockdown of these proteins does not affect the CD19 expressions, and for that they use the different clones. Out of the different clones that they have made, they have used some of the clones whose results are shown in this panel C, where we can see that the CD19 expressions are significantly higher compared to the wild type. So this CD19 expression should be enough, or enough to show enhanced cytotoxicity of CD19 CAR-NK92 compared to the NK92 wild type against the target cells that has CD19 expressions. So in this figure, they just shows the method of preparations of the different CAR-NK92, CD19 CAR-NK92 that can be used to further confirm or further analyzed to get to know which of the two mechanisms plays a major role in their enhanced cytotoxicity, whether both of them plays an equal role or one of them is better contributions towards the other one, or it is completely on the extreme side. That is, only one of them plays a role towards enhanced cytotoxicity, whereas other one has negligible contributions. That we will discuss in our subsequent videos. So I hope you understood the CRISPR-Cas9 methods that they used. Usually people clones after Cas9 positive make the Cas9 isogenic clones of Cas9 positive cells, but they make the isogenic clones after everything is done. That is, it is knocked down the individual proteins or the dual proteins are been done. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.